Modern biology defines a living organism through 5 strict criteria: complex organization, metabolism, responsiveness, reproduction, and endogenous growth. These requirements create a paradox in the lab. While a cell grows and divides from within, that internal machinery remains entirely dependent on the specific conditions of its external environment. Biological engineering co-opts this internal machinery, isolating living cells to use them as expression systems. These systems perform highly specialized tasks, synthesizing pharmaceutical proteins, replicating DNA, or modeling disease mechanisms, all within a controlled liquid growth medium. The fundamental constraint of this work is that a living cell only maintains its complex organization and metabolism when provided with an exact, organism-specific set of exogenous biochemical inputs. Culturing cells in vitro represents the precise reverse engineering of an organism's biophysical and nutritional conditions to achieve a specific biotechnological output. In this context, mastering an expression system requires treating the formulation of its media as a deliberate, calculated biochemical engineering process. Media formulation begins at the absolute biochemical bedrock. Every growth media must supply the foundational atoms of life. Carbon, hydrogen, and oxygen are primary elements, providing energy via sugars and fatty acids. Adding nitrogen and sulfur builds necessary amino acids and proteins. Phosphorus completes the set, providing the elements to build the genomic architecture of DNA and RNA. In a laboratory setting, these elements are translated into practical media constituents: energy sources, buffer salts, and mineral metals. Satisfying a cell's foundational elemental needs is the mandatory first step before attempting to force that cell to overproduce specific biological products. The simplest and most robust expression system used in biotechnology is the prokaryotic cell, specifically strains of E. coli. To manipulate these bacteria, researchers target specific internal structures: the plasmid for DNA and the ribosomes for protein synthesis. The exact formulation of the liquid media dictates how efficiently these internal structures behave. This chart shows the makeup of M9 Minimal Media on the left, a formulation restricted to basic salts and a single carbon source. M9 forces E. coli to synthesize complex molecules from base salts, creating a metabolic burden that maintains genetic strains without uncontrolled overgrowth. By contrast, rich media formulas like terrific broth use a very different biochemical strategy. Supplying pre-formed amino acids and vitamins through yeast extract and peptone completely bypasses the cell's metabolic burden. This ultra-rich environment completely removes standard limits on cellular growth, driving explosive bacterial multiplication and massive protein overexpression. However, these nutrient-dense environments introduce a major biophysical constraint: the requirement for absolute sterilization against competing microbes. Because bacterial media constituents are chemically robust, they can survive extreme physical sterilization techniques. The standard protocol involves an autoclave, which subjects the solution to high pressure and heat at 121 degrees Celsius for 20 minutes. Prokaryotic media formulation is an exercise in brute force engineering, balancing metabolic forcing with extreme physical sterilization to yield high-efficiency biological factories. Culturing higher-order eukaryotic cells requires managing a significant increase in biological complexity. Eukaryotic cells possess a delicate internal architecture, including specialized organelles like the mitochondrion, the Golgi apparatus, and the nucleus. Even intermediate eukaryotes, like yeast, require highly specific media variants to maintain productivity, such as YPD broth or synthetic defined yeast nitrogen base. In yeast culture, researchers utilize deliberate biochemical omissions, such as CSM media without tryptophan, to select for engineered strains that have gained the genetic ability to produce that missing nutrient. This complexity reaches its peak in mammalian cell culture. At this level, simple salts, sugars, and yeast extracts are no longer sufficient to keep isolated cells alive in a flask. To sustain these cells, researchers must mimic the complex in vivo environment of a living animal by adding undefined biological growth factors. A standard mammalian media, like DMEM, requires the inclusion of fetal bovine serum and precise pH buffers, like sodium bicarbonate. This shift creates a logistical challenge. The complex proteins and vitamins in animal serum are heat labile and would be destroyed by the heat of an autoclave. Consequently, mammalian media requires cold, mechanical sterilization. The liquid medium is forced through a sterilized membrane filter with a microscopic pore size of exactly 0.22 microns, physically removing potential contaminants. Once filtered, adding any remaining supplements requires absolutely flawless aseptic technique, as any oversight leads to immediate contamination. As eukaryotic complexity scales, media preparation shifts from a chemical mixture to a delicate, environmentally controlled simulation of an animal's bloodstream. The vast spectrum of cellular environments required across biotechnology demonstrates that biological domains cannot share a single universal growth medium. Every species and application necessitates its own specific biochemical profile. You see this in the difference between large-scale bacterial cultures for plasmid production in superbroth and the highly specialized requirements of eukaryotic applications, such as culturing insect tissue from Drosophila. Whether utilizing Drosophila cells in Schneider's medium or bacteria in LB broth, the biochemical environment remains the ultimate control variable. If a single factor, the pH, the carbon source, or the 0.22 micron sterility threshold is missed, the entire expression system fails. Progress in cellular and molecular biology relies on two equal halves of engineering. Mastery over a cell's endogenous genetics provides no practical advantage without the corresponding ability to engineer and manipulate its exogenous biochemical environment.